Good morning, everyone. Welcome to the Arthritis Web Seminar Series. Today is our lecture number 54 on gut microbiome and arthritis. Our guest speaker today is Dr. Tejpal Gill from Oregon Health and Science University. Before we start the introductions, just a reminder to the audience, please post your questions on the Q&A tab on Zoom. Over to you, Rob.
Welcome, everyone. It's a great pleasure to welcome Dr. Tejpal Gill to our seminar series this morning. Dr. Gill is currently assistant professor and director of the basic research in the Division of Arthritis and Rheumatic Diseases in the Oregon Health and Science University in Portland, Oregon. Prior to her coming to Portland, she completed a postdoc at Case Western Reserve and then subsequently at the NIH, where she was actively involved in mucosal immunology and gut microbiology, working with Bob Colbert and others at NIAMS. She was very productive of exploring the gut microbial immune response in the B27 transgenic rat. It's been very productive. And now she's established her own laboratory in Portland. So Tejpal, thank you for joining us at this I realize it's a very early hour in Oregon, and we appreciate you joining us. Welcome.
Thank you, Dr. Inman and Dr. Kapoor. Thank you for the kind introduction, and thank you everyone who's listening to me today. So for today's talk, I am trying to get this humongous topic in 35 or 40 minutes, where we talk about the impact of the gut microbiome in arthritis as well as beyond. And I do realize it's a little bit beyond this half an hour. So let's slice it into bite-sized pieces. I have no disclosures. So for today's talk, I will be focusing primarily the first 1/3 part on the role of microbiome in health and disease, and then we will switch gears and talk about host microbiome interactions in spondyloarthritis. And in the end, I would like to share just a couple of slides to talk about what role microbiome is playing in disease alleviation, be it in stratifying patients for precision medicine, or looking at the role of fecal microbiota transfer, or very recently articles coming on the bioengineering of microbes to help fix the gut microbial dysbiosis. So with that, let's begin with looking at the role of microbiome in health and disease. So I really like this slide that I'm starting with, and it tells us that how vast the microbiome is. So the microbiome is it's a ecological system that is living inside and on us, and each individual is harboring around 10 to 100 trillion microbes on their person, be it most of it is present in their gut. And if we look into terms of the species, there are around more than 10,000 species found to be identified living in human bodies. If we compare it in terms of the genomes, while we have around 22,000 genes, it's estimated that the microbial genes on and in us are 100 times more. And this is very interesting because there are 10 times more microbial organisms in the human body than there are human cells. So we are 10 times more microbes than we are humans. And I like the term coined, which calls us superorganisms, where the gut microbes, or the microbes on our skin or other parts of the body, and our immune response interact with each other to form and respond and this is underlying health and disease. And it's no surprise that almost 90% of all diseases now can be reached in some way, shape, or form back to the gut health and the health of the microbial communities. Looking a little bit in detail into the microbial communities, while we talk about the microbial communities, everybody is essentially just thinking or talking about the bacteria. And I just wanted to put this slide out to re-emphasize that there is bacteria, there are viruses, and there are fungi living in our gut. And while the fungi and viruses form a smaller part of the microbial component, they're nevertheless very, very important. The fungi form 1 to 2% of the microbial biomass. And in the recent years, there have been many publications which have shown that fungal dysbiosis has been implicated in many diseases, such as inflammatory bowel disease, as well as many types of rheumatic diseases. And fungal and bacteria, they have an interaction, which is called the interactome in the gut. And those interactions are altered in healthy individuals as well from patients with either gut inflammation or other diseases. And then there is virome, and it is omnipresent in the gut and in form of viruses, as well as in form of bacteriophages and latent genes in the bacteria. So they affect the bacteria in many ways by their interactions directly or indirectly, thus affecting gut microbial dysbiosis. And this slide just shows that the microbiome, even though it is an important part, it goes through many transition phases throughout our life. There is immune imprinting on the left-hand side. If you see, there's immune imprinting from the maternal microbiome as well as the metabolome. And once there, after birth, there are pioneer microbial species that are colonizing the gut, and there are many factors that decide which species colonize, be it a vaginal microbiome or a C-section, or whether the infants were breastfed or they were formula fed. And its consensus is that in the first 5 years of life, there's primary succession where the microbiome changes from an infant microbiome, largely lactobacilli, into a complex adult microbiome. In many cases where there's malnutrition or childhood diseases, you'll see that this primary succession is either delayed or it just stops, and you still keep on seeing infant-like microbiota, which is more associated with disease in older individuals. And then once the microbiota colonizes and it forms almost adult-ish variation, it's very, very stable, although insults like antibiotic treatments or infections will change it, but it will always, it's a very stable community. And this community will then start going, decreasing in microbial abundance and alpha diversity in the associated with old age and senescence. So just to add more pieces to the puzzle, microbial communities are very, very different in different parts of the body. And then perturbations in the different microbial communities is associated with distinct diseases. Now, in this slide, if you see, the actinobacteria are in the blue, Bacteroides are in yellow, and the firmicutes are more like reds and pinks, and Proteobacteria is in green. And if you see these pie charts, you can see that gut microbiome is largely made up of Bacteroides and then firmicutes, whereas the vaginal microbiome is largely made up of lactobacilli. Look into the oral microbiome, pretty much all of the microbial phyla, or bacterial phyla, are present in these. And dysbiosis of distinct gut microbiomes in our body or on our body are associated with different diseases. For example, oral microbiome dysbiosis is associated with gingivitis, periodontal diseases, and more recently it's been associated with rheumatoid arthritis. And then the gut microbiome, as we all know, has been associated with obesity, metabolic syndrome, diabetes, IBD, various rheumatic diseases, and other psychiatric disorders. And here I'm going to now focus a little bit more on the gut microbiome and rheumatic diseases. And here is a slide from Jose Scher's group, where they have just very nicely shown differences in healthy individual and then different types of rheumatic diseases: psoriasis, psoriatic arthritis, rheumatoid arthritis, and ankylosing spondylitis. And you can see that the microbiome or the microbial dysbiosis is associated with all of these disorders. It's not to say that microbial dysbiosis is causal to disease, but it is associated with these disorders. And then along with the alteration in the microbial dysbiosis, we see an immune dysregulation in these individuals, which is largely dominated by upregulation of immune and inflammatory cytokines such as IL-17, IL-23, TNF-alpha, IL-6, and a downregulation of IL-10s and 22. And further honing in on spondyloarthritis, I just wanted to briefly, before I go into the microbial perturbations in this, just briefly go through the clinical diagnosis. So spondyloarthritis, it's a group of diseases, like I just mentioned and showed you in previous slides. So have axial spondyloarthritis, acute anterior uveitis, psoriatic arthritis, reactive arthritis. And all these distinct disorders are grouped together in this under this umbrella because they have common clinical manifestations, which could be articular in nature, such as enthesitis, peripheral arthritis, dactylitis, or extra-articular in nature, that is inflammation of the eye, the gut, and the skin, as well as cardiac inflammation. Now, there is a strong association with the gene HLA-B27, which is an MHC class I molecule, and its function is basically to present self as well as microbial antigens to the T cells. So before going into why do we even suspect that there is gut microbial dysbiosis in spondyloarthritis, when we started this research, almost 10 years ago, we were more focused, gut microbial dysbiosis was more associated with IBD. It was just starting, and we found that there is a huge overlap between spondyloarthritis and IBD. Now, 50 to 70% of patients with axial spondyloarthritis will have subclinical gut lesions, and 6 to 10% of those will go on and develop overt IBD. On the other hand, 50 to 80% of IBD patients will have arthritis. And the other evidence that gut microbiota plays a role in spondyloarthritis and other rheumatic diseases came from experimental rodent models. And these models were highly, highly dependent on the gut microbiota for their disease development. For HLA-B27 transgenic rats, raising them in germ-free they will not develop either arthritis or gut inflammation. Similarly, the SKG mice, the germ-free rats will have attenuated arthritis or ileitis. A little bit more detail on the rat model, since we'll be talking about it in the next couple of slides. So as you can see in germ-free rats, there is this is the histology of the colon, and it's really nice, and you can see the epithelial monolayer. And when you raise them in specific pathogen free, you can see there's evidence of colitis and presence of inflammatory infiltrates. So these animals develop spontaneous colitis as well as peripheral joint inflammation. And for these disease, the gut microbiota is required. So when we started this project at NIH in collaboration, with Jim Rosenbaum's group here at Oregon, we thought that if we examine the effects of HLA-B27 on the gut microbiota on 3 different rat genetic backgrounds, which all have HLA-B27 at the same genetic loci, then we should be able to find common microbes that could cause or promote spondyloarthritis. So we took these 3 backgrounds, and then at around different time points, we sacrificed these rats and then performed histology and then performed transcriptome analysis on the gut, as well as the gut microbiome sequencing. And what we found was that while the DA rats do not develop any disease, Lewis and Fisher rats develop spontaneous arthritis as well as gut inflammation. And what we found in those backgrounds was, while their immune response was largely overlapping, their microbial dysbiosis there was a huge non-overlap between them. So Lewis rats had their own microbial dysbiosis going on, and the Fisher rats had their very distinct but dysbiotic community in their gut. And this led us to pursue this question to understand what's the relation between the diverse gut microbes and host immune response. And also led us to propose the ecological model of dysbiosis. So what we see here is that healthy rats, when are very, very similar in terms of their gut microbiome. They do not have any disease. And with HLA-B27, Fisher and Lewis backgrounds will go on and develop disease. And despite their common immune response, they will have very, very different microbes that were associated with that disease. So we went on and performed multi-omics and network analysis. And using those, what we discovered was, here is a Lewis clusters 1 and 4 on the left, and a Fisher interomic cluster 1 on the right. And what we found was most of the genes that were associated or correlated with these microbes were overlapping, as shown in either yellow or pink colors between the two backgrounds, whereas the microbes that are associated with these genes were totally different in the different backgrounds. And this re-emphasized the role of the microbiome, that the function of the microbiome or the microbes in the gut is redundant. And therefore we moved on and started to look. So this is just a heat map showing that most of these genes from Lewis and Fisher rats were overlapping, and they all had, when we put them into pathway generation software, we could find that these were innate and adaptive immune response genes, which were upregulated and correlate positively with different microbes on these backgrounds. So if different microbes can correlate with the same immune response, we thought maybe we need to look more into the metabolic function rather than the phylogeny of these microbes. And therefore, what we did was we performed inferred metabolome analysis using a software tool called PyCrest, and we compared them, correlated them with the histology. And what we found was, in both these backgrounds, while the microbes were different, the inflammation-associated metabolic pathways were largely overlapping, and we found perturbations in short-chain fatty acid pathways, as well as inflammation-associated pathways such as lipopolysaccharide biosynthesis, as well as steroid hormone biosynthesis. And we also found increase in bacterial chemotaxis and flagellar assembly pathways. And further proof was parallelly being done by Mark Asquith. And what he saw was that when you give these rats either propanoid or butanoid in their drinking water, the level of inflammation comes down. And this shows us that microbial dysbiosis, the effects of microbial dysbiosis can be metabolic in nature, and then which can be then treated to achieve homeostasis and disease alleviation. Okay, and I'm going to skip this slide, and then I'm going to straight move to following these results from our animal models. We've wanted to see if we see similar responses in patients with axial spondyloarthritis, and we also wanted to study host immune regulation. And what we did was we performed IgA sequencing in axial spondyloarthritis patients. And what we did here was we took the stool samples, and then since the gut makes a lot of IgA, and then IgA coats these microbes. And previous research has shown that IgA-coated microbes are cholinergic in nature. So we wanted to see if the IgA-coated microbial population is different between axial spondyloarthritis patients and healthy controls. So we took gut microbiome from patients as well as healthy individuals, and then we sorted them based on their IgA into IgA positive and IgA negative fractions, which were then sequenced, and then we performed IgA index analyses. And what we see here is that in the fecal samples, there is an increase in IgA-coated microbes in axial spondyloarthritis patients as compared to healthy individuals. And many of these microbes that are that increased in their IgA coating have been previously reported to be associated with the disease. And then furthermore, when we looked into their metabolic potential by using pie crust analysis, we found that in the fecal sample, IgA fraction is associated with increase in many of these inflammatory pathways, such as biosynthesis of unsaturated fatty acids, isopropanol biosynthesis, and so on and so forth. However, IgA negative fraction, even though same number of bacteria were taken and they had very similar alpha diversity, we did not see many metabolic perturbations associated with IgA negative fraction, highlighting the role of IgA positive fractions and, kind of narrowing down our search from all of the microbiome to then focusing into the IgA positive fractions and their interaction with the host immune response underlying disease. And then we looked into these fecal microbes, which are IgA positive, and looked into their association with disease by using the BASDI, or the disease activity index. And we found that these Firmicute family member microbes are positively correlated with the disease. So they're present more in patients as compared to healthy individuals, and also increase with the disease severity. We think that these might have an important role to play. However, the microbes which are shown to be decreased with disease abundance can be thought of as, we think that they could be like bystanders, or the commensal microbes which are decreasing, and then the pathobionts are kind of increasing with in disease. And similar studies have been done by another group where they found that the IgA-coated E. coli correlates with Crohn's disease spondyloarthritis, and also the increased production of IL-17 and increased inflammation in the gut. And I also wanted to switch gears a little bit from IgA, that is the local immune response, to IgG, which is the systemic immune response. And there's this paper that just recently came from Belcade Lab, and they were looking into the IgA sequencing like we did, but in addition, they also looked into the IgG sequencing. Now, IgG sequencing what it does is they just pair fecal samples with the plasma samples from the same study subject. And what we'll see is, since the gut microbes never have seen an IgG, if there are fractions which are bound to IgG antibodies, that means that the microbial products have somehow crossed the gut barrier surfaces, and there has been translocation and inflammation in these patients. And what they found was that patients in both Crohn's as well as ulcerative disease have a higher abundance of IgG positive microbes as compared to healthy individuals. And this is just a principal component analysis plot showing that they are very, very distinct microbial communities as compared to healthy individuals. And then they looked into certain microbes which seem to be driving these diseases in Crohn's disease and ulcerative colitis. So IgG repertoire can actually identify gut microbes that can translocate across the barrier surface and give us an idea of how the, say the gut joint axis, or how the effects of the gut microbial dysbiosis are kind of moving outside from the local response to the systemic. Coming back to our own research, what we're doing is to investigate host microbe interactions, and we're doing it through the microbial function by looking into the fecal and plasma metabolic profiling to understand. So the metabolic profile at a single time will give you an accurate picture of how the host metabolites and the microbial metabolites are interacting and the role they play in health as well as in disease. So we're looking into the local and systemic responses by fecal and plasma metabolic profiling. We're also looking into single cell expression signatures in PBMCs in various types of spondyloarthritis. And to understand host microbe interactions, we have developed colonic organoid culture systems in our lab to understand the interactions in vivo. So for the metabolomic analysis, what we're doing is, for most of these experiments, we're looking into various different types of spondyloarthritis, predominantly axial spondyloarthritis, acute anterior uveitis, and psoriatic arthritis, because we're also very interested in understanding how a common genetic marker such as HLA-B27 can predispose different individuals to different diseases, such as AxSpA, AAU, or PSA, and whether they all have different host microbe interactions underlying disease pathogenesis. So for the fecal and plasma metabolic profiling, what we're doing is we're taking stool and blood samples from paired samples from healthy individuals and patients with different types of spondyloarthritis. And then we are doing gas chromatography and liquid chromatography based mass spec, and then analyzing the data. And just some preliminary data has been trickling in, and I just wanted to share. This is looking at the metabolic profile of axial spondyloarthritis patients, shown in red, in comparison to healthy individuals, shown in green. And it's an unsupervised clustering, so we were very, very excited when most of the patient samples are clustering together. And you can see that this there is an increased metabolic increased abundance of various metabolites, and there is decreased abundance of various different metabolites in patients as compared to the healthy individuals. And when we were looking, we've just started looking closely towards them. And one of the interesting ones that I wanted to share was the deoxycarnitine and steroyl L-carnitine. So carnitines have been microbial metabolites, and they've been associated with increased gut permeability. And we have previously shown that patients with axial spondyloarthritis have increased levels of gut inflammation as measured by their fecal and plasma calprotectins. So seeing those patients, axial spondyloarthritis patients with and without acute anterior uveitis, have an increased levels of carnitines, kind of further emphasizes the point that there is increased gut permeability, which is playing a role in the disease pathogenesis. Just switching on to the single cell expression signatures. And with single cell, so single cell basically looks into the RNA, the transcriptome of each individual cells. And what it does is it kind of counteracts the caveat of bulk RNA sequencing, where you get RNA gene expression of the whole group of cells. And now, since PBMCs are such heterogeneous population, it's really, really nice to know whether the increase or decrease of genes that we were seeing, gene expression that we were seeing in our bulk RNA sequencing, is coming from whether the population has expanded in those patients, or whether the gene expression is upregulated. So to do that, the PBMCs were isolated from patients and healthy individuals belonging to various different types of spondyloarthritis. And then they are basically run through the single cell sequencing, where each cell is encoded in a single gel bead, and therefore its expression profile is defined. And then there's pseudo bulking, and we start seeing those nice UMAP populations. And here I'm just showing you one example UMAP population. Here's a healthy control and patient with axial spondyloarthritis. And you can see that there are many, many differences just in terms of their cellular phenotype. There is an expansion of NK cells and gamma delta T cells, MATE cells, and many populations of T regulatory and other CD8s and CD4s in axial spondyloarthritis patients as compared to the healthy controls. However, we saw a decline in CD8 naive population. So furthermore, we have added the cell surface epitope data into it, and we're performing site sequencing of these populations to understand how different cellular subtypes are behaving in patients versus healthy controls. And for the last part of our experiments going in the lab, we're looking into the colonic organoid system. So for this, we have developed a system to generate organoids from gut biopsies of patients and healthy individuals undergoing routine colonoscopies as well as disease-associated colonoscopy procedures at OHSU. And what we've done is, here are the organoids. You can see early forms. So organoids are essentially a small enteric structure which mimics the gut epithelial biology. So early on, you can see them as spheroids, and then as they grow on, they'll start becoming globular organoids. And here you can see nicely, it forms a tube-like structure mimicking the gut. And these are the epithelial cells, and these are the epithelial cells outside. And what we saw was that here is just looking into the HLA-B27 negative and HLA-B27 positive patient organoids. What we saw that, excuse me, HLA-B27 positive healthy individuals have a decreased MUC2 expression, which is the mucus encoding protein, and in comparison to healthy individuals. And we have done some preliminary studies on axial spondyloarthritis patients, but the data is still variable. But we have seen that with HLA-B27 positive healthy individuals, there's a decreased production, and decreased mucus generation has been associated with gut inflammation. Okay, so what we're doing further is we are trying to understand using organoids and developing them into epithelial monolayers. We are looking into the epithelial barrier. We're looking into host microbe interactions by subjecting and then altered lipopolysaccharide sensitivity by adding LPS and doing bulk RNA sequencing. So using these animal models earlier, that we have shown you that microbial dysbiosis is dependent upon host genetics. And while the microbes were different, they all were very, very similar in terms of their metabolic profile. And then also that mucosal IgA response to oral and fecal microbes was altered. And today I just showed you the fecal microbiota, but we also performed the similar analysis on the saliva and saw similar trends, but to a milder extent in axial spondyloarthritis patients as compared to healthy individuals. And these are the current studies. So to sum it up, the gut microbial dysbiosis can be affected by host immune response, genetics, as well as different markers. And the environment, diet, and antibiotics can affect its severity and kind of move it across the spectrum of inflammation. And for the last part of my talk, I'm going to, I'll be focusing on microbiome in disease alleviation. Let me just go back. Yes. Disease alleviation, and we'll be looking into drug microbe interactions, as well as looking into engineered microbes, which can fix the microbial dysbiosis caused by antibiotics, as well as end by looking into fecal microbiota transplants and oral biotherapeutics. So microbiota can process, different types of medications and then impact the effect of active components of the medication. So this is a study done by Renuka Naik from Jose Scher's group, and they were looking into methotrexate, and they've shown that it impacts the gut microbial pathway and alters the host immune response. They looked into baseline and post methotrexate treated patients, and without low methotrexate and high methotrexate, and you can see there is post-treatment, there is an altered microbial community structure in patients treated with methotrexate. And here you can see is that once you start treatment, the high doses start grouping very, very differently from the low dose and non-treated, which shows you that the microbial community is just moving or responding and changing in response to methotrexate treatment. And here is looking into the Bacteroides or abundance, and you can see there's a decreased abundance of microbes with high dose treatment of methotrexates, and these are the while the firmicutes shown in red are increasing with methotrexate. So altering the gut microbe, the medications can have an effect on altering the gut microbiome. And then another study, the gut microbiome was associated with response to methotrexate. So you can, what they did is looked into the gut microbiome of methotrexate responders versus non-responders in red and responders in blue. And you can see they are separating really nicely here. And looking into their metabolic pathways, you can see in non-responders have an increased abundance of various MAP kinase signaling pathways, longevity regulating pathways, whereas the responders have oxidative phosphorylation and various biosynthesis pathways. And when they included this information they were able to predict responders and non-responders really nicely when they included these metabolic information along with clinical information shown here in green. And this red, the area of curve is just the clinical information. So microbes can be, along with clinical information, microbes can be a good predictors of response and can help stratify patients for different treatment strategies. And here is another study where they showed that fecal microbiota signatures were associated with the response to ustekinumab in Crohn's disease patients. And you can see when they combine the clinical as well as microbial data, the area under the curve for prediction for both response and remission is highly increased than just using clinical data alone in black. And again, in looking into the anti-integrin therapy in IBD, vedolizumab, again, you see that using microbial predictors along with other clinical markers allow to predict the responders or non-responders really, really nice in multiple different studies. And here's this recent study that I wanted to talk to you about. It's engineering of live microbes for the prevention of antibiotic-associated dysbiosis. And what happens is in a normal gut, when you give ampicillin for an infection, it also alters the gut microbiota, and sometimes it causes expansion of C. difficile and other microbes that are antimicrobial resistant. And what they did was they then gave patients a probiotic that is lactobacilli lactis. What it does is it produces beta-lactamase. Now, beta-lactamase breaks down penicillin into an inactive ampicillin into inactive penicillinoic acid. And what it does is then it inhibits the gut microbiota disruption and inhibits the formation of antimicrobial resistance, and also inhibits the Clostridium difficile expansion. And this is really, really great because, antibiotics have such adverse effect on the gut microbiome and then other diseases. So this is a really, really nice way and a nice study to show that giving probiotics, which are engineered to target specific genes, can restore a healthy gut microbiome. And for the really last two slides, I'm looking into the fecal microbiota transplant. And what it is essentially is that giving fecal microbes from a healthy individual. Now, the hardest part, I think, here is to define a healthy individual, but healthy individual gut microbial community is given as either lyophilized pills or enemas to patients with different types of disease for their disease amelioration. And here I've just listed a few of the fecal microbiota transfer trials that are going on that I could find on the clinicaltrials.gov website. They're looking into ulcerative colitis, they're looking into chronic inflammatory diseases, and they're looking to various different forms, pills versus fecal microbial slurries. And here is just a recent study where they were looking into the safety and efficacy of fecal microbiota transplant for psoriatic arthritis. And while this study, what they did was they just randomized people to receive either fecal microbiota transplant or a sham, and then they looked into their treatment failure, and you can see like if for 26 weeks, and then they looked whether they needed extra treatment. And while fecal microbiota came inferior in this is really a small number of study, and they did not define their healthy individuals. So even though this study doesn't work, I still believe that fecal microbiota transfer could be a way to go. And since with fecal microbiota transfer, there is lyophilized, there's a way to give lyophilized microbes in a capsule. And this study that just came in looked at preparing these bacteria with a mineral coating on the bacterial surface. And what it does is once they are mineralized, when ingested, they can withstand gastrointestinal insults, and the mineral coating is neutralized and it's released once it reaches the bile acid. So it's a much better way, but it's just a paper that came in. I think more studies are needed, but I thought it was an interesting read to look how these bacteria can be mineralized to prevent the gastrointestinal insults, and thereby increasing the effectiveness of fecal microbiota transplant. So in the end, I just wanted to sum up with this slide. It shows that microbiome affects us in various different ways. And in patients, you'll see disease-associated dysbiosis, altered immune response from the host, and then altered metabolic perturbations from host, as well as the microbes, which may be underlying, and their interactions might be underlying disease pathogenesis. And then we looked into the effect of diet on diet modulates the microbiome in the gut, so therefore can move across towards inflammatory or as healthy gut. And then we looked into the drug efficacy, fecal microbiota transplant, and also looking into how we can generate engineered or bespoke bacteria to promote gut homeostasis. So with that, I'd like to thank you. I'd like to thank my mentor, James Rosenbaum, Bob Colbert, and all my collaborators and past and present lab members, as well as the funding sources. Thank you so much.
That was a terrific overview. Thank you so much.
Thank you.
Just a reminder to the audience, please post your Q&As on the Q&A section on Zoom. Dr. Gill, just starting off with the gut microbiome research which you're doing, and also the integration of other omic strategies like single cell, looking at metabolomics. Could you just summarize, how do you plan to actually bring this all together?
Yes.
right?
So
Because you're looking at metabolites, you're looking at things from a single cell perspective. Do you think the cellular constituents at the local level or even at the systemic level would contribute to systemic changes? Maybe could be metabolites, but then eventually impacting the gut microbiome. That actually help us stratification of patients or identifying disease pathogenesis.
yes. So you're essentially asking me to sum up my research. So what I think was, so we have this patient population where we have largely focusing on 3 diseases: axial spondyloarthritis, acute anterior uveitis, and psoriatic arthritis. And because they're all B27 associated diseases, and I'm very interested in understanding what is the difference between the disease and also within an individual disease. The idea is we find these studies where they say, oh, there is gut microbial dysbiosis, but what does it do? Because gut microbial function is so redundant. If somebody else performs those same studies in a different part of the world, then they'll come up with different microbes. So using microbes as biomarkers becomes really hard to kind of consolidate and understand, because in another part of the world, they might not even have those microbes in the gut. And therefore, we wanted to understand that, okay, these are the microbial perturbations. And in the same patients with these microbial dysbiosis, are there any metabolic perturbations, right? So does it lead to any metabolic perturbations? So we are doing all these paired microbiome metabolome analysis from the same patient so we can perform correlations and understand. And then when we were looking into the gut microbiome, I'm very, very interested in understanding whether there is local response, and then is there a systemic response, and is there a systemic response different between, patients who have acute uveitis rather than axial disease or gut disease. So we're looking into that. And if there is a systemic response, then are there any biomarkers that we can do? How is the host responding to those systemic responses? So doing all these studies in many, many overlapping individual sample cohort will allow us to basically answer these questions, that if the microbial dysbiosis is actually translating into metabolic changes. And, it could be that there are many, many metabolic microbial changes, but they won't translate to metabolic perturbations. And which changes, when translated, will also kind of elicit a host immune response. So doing them in same patients will allow us to answer those questions and kind of link different parts together. And that's the aim of and kind of hope of this big picture study that we're doing. And then we have a rat model where we would be testing various metabolites that we are seeing some promising metabolites that we want to test in the rat model and look at disease amelioration.
Great, thank you, Tejpal. Amit Sandhu raises the question: high-dose methotrexate can also cause GI toxicity, which may be due to polyglutamate levels, but what is the role of the microbiota on these polyglutamate levels? Can the microbiota affect the influx and efflux of methotrexate and therefore rheumatoid arthritis?
So I have to say that I don't know what's the effect of microbiota on polyglutamate levels, but microbiota, and I can check this out and reach back to the in an email, but
Sure.
So microbiota can break down methotrexate, and then certain microbiota can do that more than others, and then that alters the bioavailability of methotrexate, which is actually active, and then leads to disease alleviation. So depending upon the gut microbiota, it could be that different patients, even despite they're getting the same levels of methotrexate, be it a high dose of methotrexate even, they might not be getting same active methotrexate for their disease, and therefore you see different types of responders and non-responders. And that's where I think microbiota can be used as a marker to understand whether, oh, these patients, and in this case where in Renuka's study, what they found was that it is kind of associated with dampening of immune response, which might play a role in the disease.
Great, thank you.
Question is by Paramir Kaur. Amazing presentation, Dr. Gill. We know from past studies the role of FUT2 gene in gut microbiota. Do you think it would be essential to study this gene in relation to arthritis, or could have a possible role or association with arthritis?
Could you say the gene again?
The
FUT2.
Oh, yes. So, yes and no. And I say this because it just depends. Different genes will have, the gut microbiota depends so much, the gut microbiota changes so much between individuals, such as there's so much inter-individual variability. I think the FUT2 gene is a good marker, but I really don't know how it's going to translate if we look into in terms of the either arthritis or IBD.
Great. Margaret Atkins says, excellent presentation. If I understood correctly, some bacteria cause an IgA immune response. And the question is, what happens with this type of bacteria in IgA deficient patients?
Yeah, so this is a very interesting question. There was a study where they looked into patients who are genetically deficient on IgA, and their gut microbiome wasn't, their gut microbiome was different, but it didn't seem like there was so much an effect in terms of having more inflammation. And they thought that in absence of IgA, IgM was kind of overexpressed and playing a role. So the role of the gut microbiome, IgA, is historically it's thought that IgA would target these gut microbes, which are cholitogenic in nature, and then these will be either, they're targeted either for complement or macrophage associated degradation or destruction. So and recently there have been studies where it's thought that certain species of Salmonella and other microbes can, they reach out very, very close to the mucosal layer, and they almost like seek out IgA coating. And when they're IgA coating, they can be close to the immune cells and interact. So in some cases, the microbes have found a way to, it's also almost thought to have it as a protective effect. But historically, it's thought that IgA targets cholitogenic microbes for destruction, but that IgA is large. It's very, very, it's not specific. It's kind of quasi-specific. So it targets a wide variety of microbes.
Next question, Dr. Gill, have you looked into whether if there are any alterations in the T cell receptor repertoire in PBMCs for exospondylitis patients with microbiota dysbiosis?
Yes, so we haven't really looked into TCR repertoire sequencing. We haven't done that, but there's a recent study that came from a large multi-lab cohort coming from the Garcia lab, and Michael Paley was one of the authors on it. So they have looked into the T cell receptor repertoire, and they found that certain sequences are in response to certain TCRs in response to host as well as microbial peptide sequences were expanded from the PBMCs, the synovial fluid, and also the ocular fluid of patients with axial spondyloarthritis and acute anterior uveitis, respectively. So there are recent studies which are looking into TCR repertoire sequencing, and they're finding the increase in those T cell receptors, expansion of those T cell receptors in patients, but we haven't done that so far.
The methotrexate clinical response story is very interesting, Tejpal. You highlighted it very nicely. Do you recall whether that was the methotrexate was given orally or by injection?
I'm going to say orally, at least in one study, because I put 3 studies. And, you know, because then you can look into the gut microbial dysbiosis more. Right. If it was injection, then, you know, you wouldn't see so much of an interaction. So I'm just going to say orally, but I'll check and get back to you.
No, it's, I mean, as we move into, we've been, of course, using most of our biologic treatment in AxSpa has been systemic by injection, but we're moving into, with the JAK inhibitors, we're moving into oral biologic era. So it'll be interesting to see whether it changes, there's a difference. I assume that we don't have good, I don't think we've got very good comparable data, like the profiling the methotrexate response, non-response by the gut microbiota. We don't really have good data on, say, TNF or IL-17 responses with the same analysis, correct?
I think there was a study, and I have a feeling that it kind of came either from Jose Scher's group or a collaborative study where they were looking into TNF and IL-17 responses and gut microbiota, and they found that both TNF and IL-17 alter the gut microbiota. TNF inhibitor more so, and I apologize, I should have put that paper here.
No, that's fine.
TNF inhibitor more than IL-17, but they both had distinct microbial perturbations associated with it.
Right.
But there has been a study on both, and the same paper looked and compared TNF and IL-17 responses.
Yeah, it's still a very perplexing story why IL-17 inhibitors don't really work in Crohn's disease, but they work in axial SpA, and the IL-23 inhibitors are exactly the opposite.
Right. So I think at the end of the day, axial spondyloarthritis, there's a trigger, and then there is like a perpetuation phase. And I think IL-23 is really, really important for the trigger phase, at least in our. So when we looked into our rats and their transcriptomics while they were growing, we did a spatial temporal analysis. Really, really young animals at around 1 to 2 months of age, they'll have this increased in IL-23. And as they grow older, say 3 months or 6 months, the IL-23 increase kind of went back. So there was no difference between the individuals. However, then the IL-17 just takes off. And so we think that IL-23 is important really in the trigger phase. And then once IL-17 is there, it just kind of takes off in perpetuation and inflammation. And IL-17 is also responsible for preserving the gut lining homeostasis. So, you know, patients who have axial spondyloarthritis may actually, with perturbations in their gut, might actually have worsened colitis if, you know, treated with IL-17. And there have been some studies showing that, and maybe that's the reason it likes, you know, IL-17 is good for psoriasis, you know, it works better there, but not for colitis, where, you know, the gut,
Right.
We're right on time. Thank you so much for joining us. It's an amazing, amazing overview. Thank you.
Thank you for the opportunity. And thank you for everyone listening and their questions. Please feel free to reach out.
Thank you. Bye-bye.
Bye.